Okay, hi everybody, this is Lilith and I'm one of your IAs for BIPN 160. This is my review video for lecture 1 and 2. I will go ahead and provide some detail in topics that might need some detailed explanations for people who may have not taken any neuro-specific classes previous to this one. So if you're comfortable with the detail, you can always speed up this video or jump ahead, but the detail is there for individuals who would like it. Okay, let's go ahead with lecture 1 and then we'll jump into lecture 2. So in lecture 1, we started talking about the historical scientists who laid out the foundation for neuroanatomy. The first scientist was Schwann, and Schwann proposed the cell theory, in which he said that the brain is not made up of individual cells like the rest of our body. So he basically believed that the brain was a chunk of tissue that did not have individual cells. Then came around Golgi, and Golgi proposed the reticular theory. In the reticular theory, he explained that the brain is made up of one big network of fibers. However, they are not individual cells. So he thought that there's a bunch of fibers like this, and it's just one big network that just is connected to one another. However, he did not believe that these were individual cells. Golgi also produced his own stain. It's called Golgi stain, and the stain was able to mark or stain individual neurons very sparsely, I believe about 1% of the brain. It wasn't until Cajal came around and produced the neuron doctrine, in which he actually falsified what Golgi and Schwann theorized, and he did say that the brain is made up of distinct neurons, where each neuron is polarized in a way that the signal which travels from neuron to neuron is always unidirectional. Okay, and to do this discovery, he used the Golgi stain that Golgi had produced. Okay, so these are the three names, their theories, and what the scientists believed at the time that you should know. Okay, then we jump into different types of neurons. We have unipolar neurons, which generally exist in development only and are very rare in adulthood. Unipolar neurons have a soma and only one projection, one axon coming from the soma, and because it is one projection, this is called unipolar. Pseudo-unipolar are neurons that are in the sensory nerves, and you will learn more about this in the coming lectures. Why is this called pseudo-unipolar? Because from the soma, we again have only one projection. However, this one projection splits into two, so this is kind of a pseudo, right? Fake unipolar. The bipolar neuron, pretty self-explanatory, right? It has a soma and it has two projections coming off of the soma, hence bipolar. And the bipolar neurons can be found in the retina of the eye and also the nose. Both of these we will discuss further into the quarter. And the last type of neuron that we covered is the multipolar neuron. This is the most common neuron, and it has a soma, many projections of dendrites, and one axon. Okay, hence multipolar. Okay, most motor neurons are multipolar. Okay. Now let's move on to talking about action potentials and the neuron. This is where I'll go into a little bit of detail for the action potential for individuals who would like to know a little more detail. So over here we see a neuron, a multipolar neuron to be exact. The axon of this neuron has cell membranes, right? This and this. These are the membrane boundaries. So if we zoom in into here, right, from the axon, this are the membranes of the axon. And embedded in the membrane, we have different ion channels or different ion pumps, okay? And there are 3 channels that you should know about. The sodium-potassium channel. This channel allows 3 sodiums to come out, 2 potassiums to come in. We have voltage-gated sodium channels. These allow for sodium to come into the cell. And then we also have voltage-gated potassium channels that allow for potassium to come out of the cell. Okay, now that you know the distinction between all 3 channels, let's talk about how each one of them work. And we're going to look at this graph right here. When the cell membrane of the neuron is not experiencing an action potential, it is at its resting membrane potential, meaning the charge inside of the membrane sits at about -70. So it's a negative charge, resting membrane potential. Okay? Then, once a local potential, so once some positive charges flow into the neuron from the soma, the inside of the membrane becomes more positive. And as soon as it hits this threshold, the voltage-gated sodium channels start to open. Once they open, a lot of positively charged sodium flows into the neuron cell, and this graph starts to rise. And this rise is called depolarization. Okay? Once we hit the top of this graph, what happens is the voltage-gated sodium channels close right away. So no more sodium is coming into the cell. Right at this top portion of the graph, the voltage-gated potassium channels suddenly open. And when they open, potassium is rushed out of the cell. And imagine if we rush out positively charged ions out of the cell, then the inside of the cell will become more negative. So as potassium channels are opened at the top here, the membrane is becoming more negative, and we get the downfall of this graph, which is called repolarization. The repolarization lasts a little longer because the voltage-gated potassium channels don't close until a little time has passed by. So this big dip below the resting membrane potential is called the hyperpolarization. Okay? As soon as the hyperpolarization ends, the membrane comes back to its resting membrane potential, and now the cell has completed 1 round of an action potential. Okay? You don't need to know any of these details. I believe Dr. Root said that he posted a graph of explanations of what happens in depolarization, repolarization, and hyperpolarization, but I just went through it to give you a better understanding of what exactly an action potential is and what is happening in the cell membrane of the axon. Okay? So when action potentials keep on happening on the axon, right, the signal travels all the way down the axon to the axon terminals. And at the axon terminal right here is where different vesicles full of neurotransmitters are released into this synaptic space. A synapse is the area between where 2 neurons are interacting with one another. Okay, we will talk about the different types of synapse shortly. So once neurotransmitters are released into the synapse, the molecules, which are neurotransmitters, right, they're molecules, they will look for a receptor to bind to on the postsynaptic neuron. Okay? there are 4 types of synapses. Axo-axonic, where the axon synapses with another axon. Axosomatic, where the axon terminal synapses with the soma of another neuron. Axodendritic, which is, I believe, the most common synapse type, where the axon terminal synapses with the dendrite of the next neuron. And dendrodendritic, where 2 dendrites interact with one another. Okay? Once these neurotransmitters are released into the synaptic cleft, as it is called, they are looking for receptors that they can bind to so that they can somehow affect the postsynaptic neuron, right? Either activate or inhibit it. There are 2 types of receptors, ionotropic and metabotropic. Ionotropic receptors are the receptors that allow for different ion flow into the neuron, and depending on whether those ions activate or inhibit the neuron, we will get activation or inhibition of that signal. Okay? And then metabotropic receptors are receptors that, instead of allowing ion flow to happen, they allow for some kind of mechanism to happen once the neurotransmitter binds to it. Okay? Now let's talk about the glial cells. The glial cells are the cells that are not neurons, okay? So the brain is made up of neurons and also other cells that are called glial cells. Let's explore the 5 types here. So astrocytes are the glial cells that are in the CNS, the central nervous system, which is the brain and the spinal cord. They mainly hang around in the blood-brain barrier where they regulate blood flow. They regulate metabolism of neurons and the brain. And they also buffer the extracellular environment, as in they probably excrete some components into the extracellular environment that buffers it, right? Not too acidic, not too basic. Oligodendrocytes are glial cells that make myelin in the CNS. Okay? I guess they do have some synaptic potential, but they're not well understood, but I don't think you really need to know this part. Just know that they make myelin in CNS. Schwann cells are glial cells that are found in the PNS, and they make myelin there, and they also reuptake neurotransmitters. Okay, and PNS is the peripheral nervous system, which is everything else other than the brain or the spinal cord. Okay. Microglia are the glial cells that are that have immune system properties. So they are macrophages that flow in the brain and the central nervous system. They engulf and destroy pathogens, foreign material, tissue that has died and doesn't need to be there anymore. And they are also very mobile, and they can go to different places in the tissue that has had damage. Ependymal cells are the last glial cells we discuss. They line the brain ventricles and the central canal of the spinal cord. The ventricles and the spinal canal is where we have cerebral spinal fluid flowing. Okay, ependymal cells line those areas, and I believe they're the ones that form cerebrospinal fluid, but don't quote me on that. Just know that they're here. Okay. Another thing that you should know for this class, and most likely other classes that you encounter, is how the brain is talked about in terms of location. So the front of the brain is anterior, the back is posterior, the top superior, the bottom inferior. Caudal means tail in, don't know which language, but it means tail in a language. maybe Latin, I don't know. Rostral means head. So the front of the brain is rostral, and as we go down the spinal cord, remember that if we keep going down the spinal cord, we will hit the tailbone, so to say, right, of the person, the tailbone. So that is why this end is called caudal, because it's going to the tail. Okay. And what else did I miss? And the front portion over here is called ventral, and the back is called dorsal. Okay. I went ahead and pulled these questions from the end of the lecture, and I'll go over them right now and highlight the answers for you. I think it would be best if before I say the answer, you go ahead and stop the video and just check yourself. Make sure that you know what I'm about to say, or I hope I don't make a mistake. But that will be a good way of quizzing yourself. So let's go. Which type of glial cell makes myelin in the CNS? Stop the video if you'd like. If you did or if you didn't, I would say the answer is B, oligodendrocytes. Make myelin in the CNS. Number 2, which of the following is the main excitatory neurotransmitter in the brain? And I believe it is GABA. One function of astrocytes is regulate blood flow. Let's go over the rest of the answers also to make sure that it's good practice. So make myelin, well, astrocytes don't make myelin. The 2 glial cells that make myelin are oligodendrocytes in CNS and Schwann cells in PNS. Destroy pathogens? No, because microglia are the ones that destroy pathogens. Relay sensory information? No, it's probably just talking about neurons. Okay, astrocytes don't have sensory information relay properties. Okay, this question right here is exactly the same as the top here, but I'll go ahead and write it in case somebody's following with my notes. We can use black. So we'll have rostral here, caudal here, right? Anterior here, posterior. We will have superior, inferior. Oh. I feel like I spelled that wrong. Inferior, maybe? Oh no, bad spelling. And also this is dorsal, right? And this is ventral. Okay. Oh, and last question here. What is the neuron doctrine and who stated it? Neuron doctrine was stated by Cajal, right? And Cajal said that the brain is made up of individual cells, right? They are polarized, travel the signal unidirectionally, and he also used the Golgi stain. Okay. Over here, I've compiled the learning objectives for you. I won't go over them, but you should go over them, as this could be good practice for the exams. And if you have any questions, I can always answer them over email or during office hours. Monday at 7 p.m. Okay, I'll go over lecture 2 now. In lecture 2, we started talking about some techniques that are going to allow us to study the brain in 3 different ways. The first way is to visualize the anatomy of the brain. Okay, so that's 1. 2, number two is going to be for us to visualize the neural activity of the brain. Okay, and number three is are techniques that are going to help us manipulate the neural activity of the brain. So I will make a little sketch over here on the side. Let's do it in blue. So 1, anatomy. 2, activity. And 3, manipulation. Manipulation of activity. Okay? So let's go ahead and talk about dyes. And dyes are the first way that we can visualize the brain. Oh, I'm missing an E over here. Hold on. Okay, sorry. I think it's this color. There we go. Visualize the brain. So the first stain that we talked about was the Golgi stain. Remember that the Golgi stain stains about 1% of the brain. It stains individual cells, and the stain is very sparse. Okay, the Nissl stain looks a little purple or bluish. It binds RNA, and it shows cell densities. So the Nissl stain is used to see where cells are more compact in that area of the brain. Okay, acetylcholinesterase is used to label acetylcholine synapses, and this is good for checking for brain boundaries. For example, different areas, there's clear distinctions between where one part of the brain begins and another part ends. Okay, that's what I mean by brain boundaries. And then DAPI is used to see boundaries and cell densities. Okay, DAPI binds DNA. Easy for me to remember D with DNA. An important concept with DAPI is that DNA exists only in the nucleus, and the nucleus exists in the cell body of the neuron. So DAPI, when used, will only stain the gray matter of the brain, because the gray matter is where the soma is, and the white matter is where the myelinated axons are. The gray matter also includes synaptic clefts, dendrites, and I guess any other extracellular areas of the brain. Okay. Then we talk about immunohistochemistry, and this is another way of visualizing different proteins or different structures in the brain. There are two types of immunohistochemistry technique: the indirect and the immunofluorescent. The indirect immunohistochemistry is when we have a protein that we want to target, and we form an antibody that attaches, that is specific to this protein. Then we introduce another antibody that goes ahead and attaches to this first primary antibody. And now the secondary antibody has an enzyme on top of it that takes different molecules and turns them into a different color that we can see. Okay? Meaning, if we see this color, then we know that this protein exists in the brain. Okay? Immunofluorescent immunohistochemistry is when we have a protein of interest. We form an antibody against it, and this antibody is attached to another secondary antibody, and this secondary antibody now has a fluorescent tag on it. So I'll draw a star. So instead of this being an enzyme that produces something fluorescent, this molecule in itself is fluorescent. Okay, so these are the distinctions between indirect and immunofluorescent immunohistochemistry. Okay. Then we talked briefly about in situ hybridization, and this is when if we have a gene of interest that we would like to target in the brain, what scientists can do is they can form a synthetically made part of a gene that we can maybe call a probe, and this probe has immunofluorescence or is tagged in some way that produces light. And the most important thing is that these are compatible with one another. So once this is introduced, it goes ahead and binds this gene. And because we know that they are going to hybridize, wherever this immunofluorescence comes from in the brain, we know that this one is also present in that same area because they hybridize with one another. Okay. Then we went ahead and talked about tracers. So there are 3 types of tracers: anterograde, retrograde, and some other ones. Anterograde tracers are tracers that take up the tracer in the cell body and they move it along the soma and the axon all the way to the axon terminal. Okay, so in a sense, they show, let's write it in black, they show the output of a brain area. Retrograde tracers are tracers that take up the molecule at their axon terminal and they move the tracer along the axon all the way to the soma, to the cell body. Okay, and in the same sense, retrograde tracers show inputs to a specific region. Okay, the other types of tracers, such as di-I and dextran, they basically dye the whole cell, the whole neuron. So they're not taken up at the cell body or the axon terminal distinctively. They just dye the whole neuron. Okay, but they're still considered tracers. So anterograde tracers include FAL, WGA, AAV. AAV is in a different color because this is a viral anterograde tracer. Retrograde tracers include chlerotoxin B, Fluorogold, and retrobeats, and it also includes rabies virus and retro AAV, and these are also in a different color because they are viral tracers. We will talk a little more about the rabies virus as a retrograde tracer in the future. Okay, now let's move on to genetic tools and modifications that allow us to again visualize the neuroanatomy of the brain. So the first protein or tool that we talked about is the GFP. GFP stands for green fluorescent protein. And the way GFP can be used is the scientists, or I guess in this case the geneticist, would take a promoter of a gene that has the gene X right after it, and they will attach the GFP gene right at the end of gene X. This way, when the promoter drives the production or the expression of gene X, it also inevitably drives the production of GFP. So we get protein X plus GFP. Because GFP is a green fluorescent protein, it will fluoresce in whichever area this protein X travels to, right? Because GFP is simply attached to it. So wherever protein X goes, GFP goes. So we can see where the protein moves to in the brain. So that is one way of using GFP. attaching it at the end of gene X. The second way of using GFP is if we completely take off gene X, that can also work. We don't need to produce protein X if we don't want to. We can attach the gene of GFP at the end of the promoter, and the promoter will drive only the expression of GFP. And again, because this is fluorescent, we will be able to locate it in the brain. Okay, so hopefully the logic and the understanding of how GFP works and how the promoter is able to I lost the word that I wanted to say, how the promoter basically activates the expression of whatever is after it. Okay, so keep that process in mind, because when we go ahead and talk about the Cre-Lox-P system or the TTA system, it's basically the same idea. Okay, so let's talk about Cre-Lox-P. So Cre-Lox-P, we take the gene for Cre and we put it at the end of gene X. I believe we can also just put it after the promoter. We don't need gene X, I think. So what happens? This promoter drives the expression of protein X plus Cre, right? And what is Cre? What is the protein Cre? It is an enzyme. I've drawn a generic looking enzyme right here. And what this enzyme does is it is able to excise parts of other genes. Okay, and how does it do this? The scientist will place LOX-P sequences around whatever part of the gene that the scientist wants. So in this case, if there is a stop gene in the middle of this, and the scientist wants to take this away so that the promoter can drive the production of gene X easily, the scientist will place LOX-P sequences right here, and Cre will go and attach right here onto these sides and completely take out the stop sequence. After taking out the stop sequence, you see how Cre allowed the expression of gene X because it took out this sequence that stops the expression of gene X. Okay, so that's one thing Cre can do. Another thing Cre can do is, if the scientist had placed the LOX-P sequences around a particular gene that we no longer want, then Cre can come over here and take out gene X completely. This way, there will no longer be a gene X for the promoter to drive expression. So this way, Cre has stopped the expression of gene X. Okay, so there's two things Cre can do: allow expression or delete a gene and stop expression. Okay. The Cre system can be controlled by tamoxifen. If you remember from the lecture in class, Cre, once Cre goes ahead and binds to an estrogen receptors and it becomes Cre-ER, Cre-ER leaves the nucleus, right? And if Cre is no longer in the nucleus, then it can't do any of this stuff because the DNA, the genes are in the nucleus. Okay. However, if the cell is injected or is given the drug tamoxifen, tamoxifen goes ahead and binds to this complex, allowing for Cre to dissociate from ER. And now that Cre is separate from ER, it can move into the nucleus and basically do either this or this. Okay? So think of tamoxifen as a drug that controls the activity of Cre. And because Cre controls the expression of a certain gene, tamoxifen, you see how tamoxifen basically controls whether a gene is expressed or not. Okay? Hopefully that made sense. The next system that we talked about was the TTA system. And the TTA system is basically the same idea as CRE. So TTA is a transcription factor, okay? And if you have taken molecular biology, transcription factors are proteins that interact with the promoter of a gene, and when they interact, they either activate or inhibit the promoter, hence activating or inhibiting the expression of that gene. Okay, so let's look at how TTA works. A gene of interest first has to express TTA, right? We need to actually have the transcription factor. Then the transcription factor moves along and finds the promoter that it binds to, and in this case it's called TRE. TTA goes ahead and binds to the promoter TRE, and it drives the expression of whatever this gene is, right? It could be gene X. Now, this system can be manipulated or can be controlled by the drug tetracycline. Okay, it says a little tetracycline. So once tetracycline is applied to the cell, then tetracycline binds to TTA transcription factor, and once it is bound like that, TTA can no longer do this, right? It can no longer interact with TRE. And if it is no longer interacting with TRE, then TRE promoter will no longer be able to drive the expression of this gene X. Okay, so you see how the drug tetracycline is used to stop or continue the expression of genes in the TTA system. Okay. Then we talked about immediate early genes. Immediate early genes are genes that are used to drive expression, just like we were talking about, GFP being driven, right? CRE-LOX-P, TTA. So immediate early genes are genes that are used to drive either TTA or the CRE-LOX-P system to express some kind of a reporter so that we can get a visualization of the brain areas. Okay, hopefully that made sense. So let's talk about the two immediate early genes that we learned in class, CFOS and ARC. Let's talk about CFOS first. So what scientists do is they take the promoter region of CFOS gene, right here, the promoter region, and to the promoter region, they attach a gene of TTA, right, the transcription factor. Now, once there is neuronal activity, this looks like it zaps the promoter, but it's not an actual zap. It's just the CFOS promoter becomes activated when there is neuronal activity. Okay, so when there's neuronal activity, let's say I sing, right? The CFOS, or a mouse sings. We can't do this in humans. If a mouse, mice can't sing. I'm sorry, this is terrible. The mouse smells something. There you go. There is neuronal activity and CFOS promoter is activated. Okay, when it is activated, it drives the expression of TTA. Once TTA is made, in the previous picture it looked something like this, right? Then TTA will go ahead and bind to this TET-O promoter of LAC-Z. And once it binds to TET-O, then LAC-Z operon will be expressed. Okay, so you see how CFOS promoter is used to drive TTA upon neuronal activity. Once TTA is formed, it is able to activate the TET-O promoter and drive LAC-Z expression. So that's how CFOS and TTA work. Now, to inhibit this system, we use the drug called doxycycline. And what doxycycline does is it simply stops the interaction between the TTA transcription factor and the TET-O promoter. So if this is stopped, then we never express LAC-Z. Okay. Now let's talk about the second immediate early gene, ARC. Okay, so again, same thing. The scientists take the promoter region of ARC. Next to it, they place a gene of CRE-ER, and then they have the rest of the gene. Now, the significance in using CRE-ER is that, remember from previously that when CRE is attached to the estrogen receptor, the CRE is in the cytoplasm of the cell, right? It is taken out of the nucleus when it is in this complex. It is only when we add tamoxifen that CRE is able to dissociate from ER and go into the DNA so that it can do the excising of genes and whatnot, right? Now, when the immediate early gene promoter ARC drives the expression of CRE-ER upon neuronal activity, CRE-ER is formed. However, CRE is attached to ER, so we need to add tamoxifen to dissociate CRE from ER. Now that CRE is free in the nucleus, CRE goes ahead and sees these LOX-P sites and attaches to here and takes out this stop sequence. Once that stop sequence is taken out, then the promoter can easily drive the expression of this fluorescent protein. So you see now it's green. So ARC CRE-ER system is just another way where we can manipulate genetics and genetic tools to be able to express a fluorescent protein in a brain area only when we want to, right? Only when there is neuronal activity and when we've given it tamoxifen. Okay? I don't think you need to know any more detail than what I explained or what Dr. Root explained. So I hope that it made sense. Definitely practice is key. So go ahead and stop the video and try to go over explaining to yourself how these two work and see if you get it right. Okay, I will move on now to the modified rabies virus. Okay, let me quickly go ahead and see how much I have left. Okay, not that much. So the modified rabies virus, as we all know, the rabies virus is a virus, and I think it is fatal, right? So it will kill the organism. However, the rabies virus is taken and it is modified in a couple of ways to allow scientists to use this as a tool in visualizing neuroanatomy. Okay, and how is this done? I will talk about this structure in a little bit, but let's talk about this big thing. So how this is done is we take the rabies virus right here. Notice the red spiky glycoprotein on its membrane. We extract its DNA. We find the gene that produces the glycoprotein, and we take it out. Okay. Now that we've taken it out, this virus no longer has the ability of making these red glycoproteins. And because the glycoprotein is important in infecting neurons, right? The virus uses the glycoprotein to infect the neurons. That's how it infects all neurons in the organism. This virus can no longer infect neurons without the glycoprotein. However, we want the modified rabies virus to infect some neurons, but we want them to infect only one previous neuron. So if this is the starter neuron that is infected with the rabies virus, the modified rabies virus, we want this neuron to infect only its previous neuron, and that's it. We don't want this one to infect its previous one. Okay? So, to be able to do that, what scientists did is they made they made synthesized glycoprotein proteins, and they embedded it in the membrane of the modified rabies virus. So even though this virus, its glycoprotein gene was excised, and it no longer has that gene for glycoprotein in its DNA, we have synthetically placed some glycoprotein on its membrane, meaning it has the red glycoprotein, so it can infect one neuron. However, after this layer of glycoprotein is used up for infecting one other neuron, the genome of the rabies virus has no more gene G to produce more glycoproteins. Okay, so you see how the modified rabies virus, someone in my discussion said, is for single use only. I think that's a good way of putting it, right? The modified version can only infect one previous neuron by using this layer of glycoprotein, but then that's it. This neuron can't infect the one before it anymore because it has no more gene G to produce new glycoprotein. Okay, so hopefully that made sense. So that is one thing that scientists can do. They can put synthetically made glycoprotein on its membrane to allow for the modified rabies virus to infect only one neuron previous to it. The second thing that they can do is, instead of synthetically placing the glycoprotein on the membrane, they put a different protein, in this case called ENVA. And why would we want to put a new protein of our choice over here? Well, if you put a new protein on the membrane of this modified virus, that means that we can take different areas of the brain and we can genetically modify neurons in that area to express the receptor that is compatible with this protein specifically. Let's look over here. Over here, this neuron is made in a way where it expresses receptors that fit or that are compatible with the blue protein, meaning that this modified rabies virus will not attack any other neuron and will only infect the ones that have this receptor on it. So this way, scientists are able to kind of specifically attack different neurons of their choice by using this blue form. Okay? And again, the red form, when they just added synthetic glycoprotein, that is used to allow the infected neuron to infect the neuron previous to it, but only do it once, for single use only. Okay, I kind of made a summary here, maybe this can help. So this version can infect only the previous cell and stops because its genome doesn't have the gene G, because we took it out. And this type can infect the cells that have the ENVA receptor only, and this gives the scientists the luxury to infect cells of their choice by introducing ENVA receptors to the genome of certain neurons that they want to infect. Okay? Okay, great. Now let's talk about some other tools. The MRI can be used to take images of the human brain. The MRI is able to show different structures in the brain, different sizes and stuff like that, but it's just an image. Then we dive deep into the tools that are going to allow us to see activity, right? So everything, well, let me see. This is also activity, I believe. Okay, I'll come back to that. I think Dr. Root wrote a good summary at the end of lecture. You should definitely look at that. Okay, so let's get back to electrodes. So electrodes are able to record action potentials when they happen. And as we learned, if action potentials are happening, then the neurons are active, right? There's some kind of neuronal activity going on. So if on the test you're asked, how can you tell if there is neuronal activity? Just say that you will use electrodes. Okay? Calcium imaging is another way of finding out if there is neuronal activity. In a detailed explanation, calcium influxes the neuron when the neuron is about to fire neurotransmitters or release neurotransmitters, so that calcium flux comes in and it binds to a calmodulin, which is a protein. And there are some changes that are occurring. There's some proteins interacting with one another. You don't need to know that. Just know that GCAMP is able to show fluorescence, and this indicates that there is neural activity. Okay, so calcium imaging can be used when a part of the brain is active and GCAMP fluoresces. Okay, calcium imaging and electrodes, I believe, are only limited to the surface of the brain. So if we want to look at activity in deep structures, then we have to use 2-photon microendoscopy or fiber photometry. The difference between these two is that to use the 2-photon technique, the animal that we are evaluating must be fixed in place. However, photon photometry is made in a way where the animal can be moving around and having to live in its own normal lifestyle as we're collecting information from its deep brain structures. Okay. And the technique of fMRI, F stands for functional MRI, shows blood flow changes to different sides or parts of the brain, and this indicates neural activity, especially in humans. fMRI is used in humans to detect neural activity in different parts of the brain. Okay. Now we are getting into the techniques of using different tools to manipulate neural activity of the brain. Okay, there are two types: optogenetics and chemogenetics. So optogenetics are when we can use different wavelengths of light to activate different channels in the brain that we have placed into, right? So channel rhodopsin is a channel that will activate the neuron upon, I think it was blue light or yellow light. You can look that up in the lecture. I'm not quite remembering it. But channel rhodopsin will activate neurons by allowing positively charged ions to come into the neuron. And then halorhodopsin is a channel that will silence the neurons by allowing negatively charged ions to come into the neuron. So by using optogenetics, we will be able to activate or silence different parts of brains to see whether that part of the brain is required or necessary for a certain function. Just like optogenetics, we also have chemogenetics, and chemogenetics is, I believe, these are again channels, I think, or receptors, muscarinic receptors, I think, modifications of muscarinic receptors. And this one in specific activates neurons, and this one silences neurons. And just remember that because the name is chemogenetics, it means that we have some kind of synthetic molecule that is either activating or inhibiting these receptors, which in turn activate or inhibit brain areas. So chemogenetics as well as optogenetics are ways of manipulating neuronal activity in, for example, mice or frog models. Okay, not humans, because we cannot do these genetic modifications on humans. One technique that we can use on humans is the transcranial magnetic stimulation, TMS for short. And this is basically a big magnet that, when brought close to the skull of the human, allows changes in ion flow. And the changes in ion flow activate or inactivate brain areas. Okay, again, optogenetics and chemogenetics are used for not human models, and TMS can be used as treatment for humans. I took out this portion of the lecture and put it here, how to ace the exams per Dr. Root, or these specific exam questions. So 3 main types of experiments. How to find anatomy, right? So you want to use interrograde and retrograde tracers if you want to see which brain areas are connected to one another. If you're looking to see boundaries or cell densities of the brain, then you want to use some of the dyes, right? You can also use some genetic tools such as GFP or CRELOX-P to also allow for different reporter proteins to be expressed. And then you can follow those reporter proteins to see where they end up. Okay, functional activity is found by recording neural activity. For example, electrodes, calcium imaging, the 2-photon microendoscopy, and the, I believe, what was something photometry, fiber photometry for mice and other animals, and then fMRI for humans. And the third experiment is manipulation. And manipulation, think of it as turning on and off different brain areas to test causality, as in cause and effect, right? If I silence this brain area, what will happen? If I activate this brain area, what will happen? An important thing that will serve you well in this class is this. If you ask yourself, or if the test question is asking you, is this brain area required or necessary for something to happen? For example, is the occipital lobe required for me being able to see, right? Or a mouse being able to see? Then one thing you can do is silence the occipital lobe, right? I'm talking very generally. You can silence a brain area and see if that same function is still happening. If it is no longer happening, then you are correct. That area is needed for that function to happen. But if, I don't know what I just said. I think I said it wrong, but let me say it again. If you silence a brain area and the function stops happening, then you are right. That brain area is needed for that function. But if you silence a brain area and the function just keeps on happening, then you know that that brain area is not needed for that specific function. Okay, and the same goes, or the opposite goes for is something sufficient, or can something do a certain function? So in that case, you want to activate that brain area and assess what it can do. Okay, let's go over these practice questions. I think I remember the answers. I'll do my best. So if a scientist wanted to label the inputs to a given brain area, which of the following could be used as a retrograde tracer? Select all that apply. So I believe fluorogold and rabies virus is a retrograde tracer. Remember that FAL is an anterograde tracer, and the Nissl stain is a stain, right? It's not even a tracer. I don't think I will read this whole paragraph for 24, but let's see where the question begins. You hypothesize that the visual cortex, I'm reading right here, that the visual cortex in the new species of actually receives olfactory information from the olfactory bulb rather than visual information for the lateral geniculate nucleus, reflecting an expansion of the olfactory cortex. How would you test this hypothesis? Oh, I think for this is asking an anatomy type of question, right? It's asking, is the olfactory bulb connected to the occipital lobe in this new species? So what you want to do is inject, I think, a retrograde tracer. So let's write this down. I think this is right. Please email me if it's not. Retrograde tracer in occipital lobe. And then we can see if the projections are coming from the olfactory bulb. If they are, then we know that the olfactory bulb is sending its information to the occipital lobe in this new species. Okay, how would you test whether this structure responds to odor stimuli? Here we can do some kind of neuronal activity testing, so either electrodes or calcium imaging, I'd say. Right? Let's say we are testing this in this new species or a mouse, then we will have them smell something as we are checking calcium influx by calcium imaging, or as we have some electrodes pinned into the occipital lobe. So I will say electrodes or calcium imaging. And it doesn't necessarily mention any deep structures, so we won't mention the 2-photon microendoscopy or the fiber photometry, but you can, of course, mention, if this is a structure that's super deep brain, then we can use those two topics as well. Part C, how would you test if the brain area is required for the improved sense of smell? Again, the important word here is required. So if you want to know if something is required, then you silence it. So silence by halorhodopsin. I think I'm spelling this right. Rhodopsin. Right? If we silence by halorhodopsin and this new species doesn't have improved sense of smell anymore, then we know that this structure is required. Another easy way of silencing something is by just lesioning it. I don't know if we talked about this in class, but you can do this in some cases. It's definitely an easier test answer, but I would say, to be safe, talk about using some kind of optogenetic or chemogenetic tool instead of just plain lesioning the whole occipital lobe. Okay. Now let's go to number 5. Given what you know about gray and white matter, which of the following dyes would you not expect to label the white matter? So let's talk about this. So dextran, if you remember, and dye I, so A and D, those are the other type of tracers, and so these trace the entire neuron. And when we say entire neuron, that means the axon as well, right? And the axon is myelinated most cases, so it is part of the white matter, right? So that won't be it. It's not A or D because we don't expect it to label white matter. And then the Golgi stain also stains the entire neuron. And so the only answer that's left is C, DAPI, but let's explain why. Remember that DAPI binds to DNA when it stains, and DNA is only in the nucleus, and the nucleus is only in the cell body of the neuron. And because cell body is considered gray matter, then DAPI will not label white matter. Okay, question 6. Which of the following is false about the Krelok's P system? I think this was something on your problem set also. So which is false, right? So A. Cre causes a recombination at LOXP sites. That's true. Cre is the enzyme that is able to excise parts of the gene by using the LOXP sites, right? Let's skip B because it's the right answer, so let's talk about the wrong answers. C. Cre can be used to remove a gene of interest with LOXP sites on both sides. That is also correct. D. Cre can allow expression of a gene by removal of a stop sequence. That is also correct. Those are all of the types of Cre usage that I talked about in this video earlier. So the right answer is B. Cre silences brain areas. That is not true, right? Cre is not one of the tools that are able to silence or activate brain parts. You're studying the anatomy of the dopaminergic neurons in the VTA. You want to visualize their axonal projections. You know that they are the only neurons in the brain that express the gene tyrosine hydroxylase. How could you use this genetic information to visualize these dopaminergic axons? I think we had to do, let's see, I think we had to do tyrosine hydroxylase with TTA. List genetic information. I remember it had to do, we talked about this in the lecture, so I think I will just cover this because I can't remember off the top of my head, but I will cover this in my next video most likely. But we did talk about the tyrosine hydroxylase. I just forget what other thing it used. Email me if you have questions. I can answer it after I'm done with this video and I can review it one more time. Let's see if I can do B. If you did not have any of the genetic information, how else could you investigate their axonal projections? And this one's easy, right? Anterograde tracers, because if you want to know their axonal projections, where their axons are going from that place, then just use an anterograde tracer and it will take up at the cell body and move it along the axon. Okay, here are your learning objectives for lecture 2. Next up is lecture 3, but I will cover that in a separate video. Again, my name is Lilith. If you have any questions, please email me at elverdani@ucsd. And you can also send me a message through Canvas. Let me know if you have any questions or any feedback. My office hours are Monday, 7 p.m. And also make sure to check out the IA folders at the very end of the homepage of Canvas. Each IA has their own page and folder, and we're posting either notes or my video. I will post it there, and a PDF of these notes. So hopefully that can be useful for you. And that's it for now. Thank you.